Good evening, and welcome back to Sleepy Earth Anthologies. Before we begin tonight's gentle journey, if you enjoy these slow explorations of nature, history, and the quiet wonders of our world, please consider subscribing. Your support helps this channel continue creating new stories and peaceful adventures for busy minds that require restful evenings. Now, settle in, get comfortable, and let the world drift softly by. Tonight, we're going to a shallow lagoon on the western coast of Australia, Hamelin Pool, inside Shark Bay, at the edge of one of the most inhospitable coastal environments on the continent. Hot, hypersaline, and exposed to a desert sky. It is the home to organisms that have been building structures on this coastline in essentially identical form for 3.5 billion years. Take a slow breath. Let your body settle. We're going a long way back. Shark Bay sits at the westernmost point of the Australian continent, where the Indian Ocean meets the edge of the Gascoyne bioregion. The Foray Sill, a shallow submarine ridge cutting across the southern entrance to the bay, restricts water flow enough to allow evaporation to concentrate the salinity of Hamelin Pool to nearly twice that of normal seawater. Hypersaline conditions like this exclude most marine grazers. Sea urchins, gastropods, fish species that would otherwise consume the microbial mats. The salt is a wall. Behind that wall, on the limestone floor of the lagoon, grow stromatolites. From the shore at twilight, they look like dark, rounded boulders, smooth, uniform mounds rising from the shallows, half-submerged, water-slicked, unremarkable. A tourist who didn't know what they were looking at would photograph them briefly and move on. What they are looking at is the oldest living ecosystem on Earth. The structures in Hamelin Pool are built by the same biological mechanisms, using the same biochemical pathways as the structures found fossilized in the Pilbara region of northwestern Australia, ancient seafloor formations dating to 3.5 billion years ago. The lineage of organisms building these mounds has been continuous, unbroken, and functionally unchanged for longer than multicellular life has existed. The primary architect of the stromatolite is a cyanobacterium, a prokaryote, single-celled and microscopic, belonging to a lineage that predates every animal, every plant, every fungus on Earth by a margin that makes those kingdoms seem recent. The cyanobacterial filament is a few micrometers long. It has no nucleus. Its DNA floats freely in the cytoplasm. Its photosynthetic machinery, the thylakoid membranes, is folded into dense, layered stacks within the cell interior, maximizing the surface area available to capture incoming photons. It has no flagella, no conventional motility apparatus, but it moves. It glides along solid surfaces using a mechanism that is still not fully understood, likely a combination of EPS secretion generating a reaction force and coordinated surface filament interactions. The motion is slow, deliberate, and directional, always toward light. The outer surface of the cell is encased in a sheath of extracellular polymeric substances, EPs, a complex mixture of polysaccharides, proteins, and nucleic acids. This mucilaginous matrix is not just structural padding. It is chemically active, rich in carboxyl and hydroxyl functional groups that carry strong negative electrical charges, which attract and bind dissolved cations from the surrounding seawater. Calcium ions, magnesium ions. They accumulate on the cell surface through electrostatic affinity, not through any active metabolic process. The chemistry does the work. The cyanobacterium lives in the uppermost layer of the microbial mat, the photic zone, where sunlight penetrates. During daylight, it glides upward through the mat to the surface, positioning its thylakoids perpendicular to incoming radiation. It splits water molecules using absorbed photons, releasing oxygen as a byproduct, fixing carbon dioxide into organic molecules. At night, it migrates back downward, away from the UV exposure of the surface, into the protective chemistry of the deeper layers. This daily vertical migration, photosynthesis-driven upward movement by day, chemotactic retreat by night, is what produces the laminated structure of the stromatolite. Each daily cycle deposits a thin layer of trapped sediment and precipitated carbonate over the previous surface. The layers accumulate. The dome grows upward, millimeter by millimeter, year by year, century by century. The cyanobacteria are not the only inhabitants of the mat. The stromatolite cross-section reveals a precisely stratified community of 3 metabolically distinct lineages, each occupying a chemical niche determined by the oxygen and light gradients of the mat's interior. Immediately below the photosynthetic cyanobacterial surface layer live the purple sulfur bacteria, chromatium and related genera. These are anoxygenic photosynthesizers. They use light energy, but instead of splitting water, they oxidize hydrogen sulfide, producing elemental sulfur as a byproduct rather than oxygen. They occupy the thin zone just below the oxygen-producing surface layer, where light still penetrates, but where the oxygen concentration drops to near zero. Their pigments absorb wavelengths the cyanobacteria don't use, filling the spectral gaps in the light budget with remarkable efficiency. Deeper still, in complete darkness, the sulfate-reducing bacteria operate on an entirely different metabolic basis. They are strict anaerobes. Oxygen is toxic to them. They consume the organic matter that falls from the layers above, using dissolved sulfate as their terminal electron acceptor instead of oxygen. The sulfide they produce as a metabolic waste product diffuses upward to feed the purple sulfur bacteria above. What is waste to one tier is fuel to the next. These 3 metabolic zones, oxygenic photosynthesis, anoxygenic photosynthesis, sulfate reduction, stack in order of their oxygen tolerance and light requirements. The physical geometry of the mat itself creates and maintains these chemical gradients. The arrangement is not coincidental or competitive. It is a self-organizing chemical ecosystem where each layer's waste products are the next layer's inputs, and where the structure of the stone determines who lives where. This is what ecologists mean when they describe the stromatolite as a biogeochemical system rather than simply a community of organisms. The biology and the geology are not separate. They are one structure. The process by which living biological activity is converted into permanent stone is called organomineralization, and it is what makes the stromatolite unique among living structures. The EPS sheath of the cyanobacterium is the primary mineral precipitation site. As the photosynthetic layer fixes carbon dioxide during daylight hours, the local pH rises. Carbon dioxide removal from solution shifts the carbonate equilibrium, increasing alkalinity. This rise in pH causes calcium carbonate to become supersaturated in the immediate microenvironment of the cell. The carboxyl groups on the EPS surface provide nucleation sites, locations where the crystallization of calcium carbonate can begin. Aragonite and calcite microcrystals begin to form on the cell sheath. Not because the cell is directing the process, but because the cell's chemistry has created the exact conditions for spontaneous mineral precipitation. The cell is literally building stone around itself as a byproduct of photosynthesis. This happens continuously at the matte surface. The newly precipitated carbonate binds to sediment grains swept in by tidal currents, cementing them in place. The EPS matrix traps additional particles mechanically. The result is a thin, cohesive layer of biologically influenced carbonate. Not quite rock, not quite biology, but a hybrid of both. The next day's upward migration of cyanobacteria passes through and above this cemented layer, leaving it behind as a permanent geological record of yesterday's surface. Over decades and centuries, thousands of these daily layers accumulate. The interior of the stromatolite is a stratigraphic archive, each lamination a day, each centimeter of growth, a century of biological activity frozen in mineral form. The fossil stromatolites of the Pilbara, structures now buried in ancient rock, exposed by erosion on the desert surface, are the same architecture, formed by the same process, preserved for 3.5 billion years. The intertidal zone of Hamlin Pool is not a stable environment. Tides expose the upper matte surface to air twice daily. In a desert climate, this exposure is lethal to most organisms. Rapid evaporation, intense UV radiation, temperature swings from midday heat to cold desert nights. The cyanobacteria have survived intertidal exposure for longer than any other organism on Earth, and the mechanism is the EPS matrix. When desiccation begins, when the tide withdraws and the air begins stripping moisture from the matte surface, the EPS sheath changes character. Under increasing osmotic stress, the cyanobacteria upregulate EPS secretion. The polysaccharide matrix thickens, forming a dense, cross-linked gel layer over the cell surface. This gel has a very high water retention capacity. The complex polymer network traps water molecules through hydrogen bonding, slowing evaporation dramatically. The outer surface of the mat dries first, forming a hard protective crust that shields the cells below from the full force of the desiccating air. The critical point is the delay this creates. The outer mat sacrifices its moisture first, buying time for the deeper cells to execute their own protective chemistry. As cellular water content drops below a threshold, genetic cascades activate. Genes encoding heat shock proteins, compatible solute synthesis, and DNA repair enzymes. The cellular machinery doesn't shut down in disorder, it executes a programmed, orderly transition into dormancy. Non-essential pathways are turned off sequentially. Metabolic rate drops through several orders of magnitude. Inside the dormant cell, specialized chaperone proteins maintain the three-dimensional geometry of critical enzymes even as their aqueous environment disappears. These chaperones hold the folded protein structure in its active conformation, preventing the denaturation that would normally occur as dehydration concentrates internal ion concentrations to toxic levels. The DNA is held in compact, nucleoid-associated protein complexes that protect it from UV-induced strand breaks. The cell becomes, in functional terms, a mineralized spore, metabolically silent, physically hardened, structurally intact. Its metabolic activity drops to less than 1% of normal. It is not functioning, but it is not degrading either. The Pilbara Craton of northwestern Australia exposes some of the oldest unmetamorphosed sedimentary rock on the planet. Within the apex chert formation, rocks dated to approximately 3.5 billion years ago, are structures that are morphologically identical to the stromatolites of Hamlin Pool. Laminated, dome-shaped carbonate mounds formed by the same process of microbial mat trapping and organomineralization. These are not just similar organisms. The biochemical pathways encoded in the genomes of living cyanobacteria, the photosystem, two machinery for splitting water, the carbon fixation enzymes of the Calvin cycle, the EPS secretion mechanisms are among the most ancient and conserved sequences in all of biology. The mutations that have accumulated in these genes over 3.5 billion years are minimal compared to every other lineage. The cyanobacteria building stromatolites in Hamlin Pool today are, in any functionally meaningful sense, the same organisms that built stromatolites in the Archean Ocean. The implications for the history of Earth's atmosphere are significant. Before the Great Oxidation Event, approximately 2.4 billion years ago, Earth's atmosphere contained essentially no free oxygen. The oxygen that now constitutes 21% of the atmosphere was produced almost entirely by cyanobacterial photosynthesis. The same water-splitting, oxygen-releasing reaction happening in the thylakoid membranes of the organisms in Hamlin Pool right now. Over hundreds of millions of years, the cumulative output of global cyanobacterial mats transformed a reducing atmosphere into an oxidizing one, enabling the evolution of aerobic metabolism, complex eukaryotic cells, and ultimately every animal that has ever lived. The air you are breathing was, in its current chemical composition, manufactured by the ancestors of the organisms building stone in a hypersaline lagoon in Western Australia. When the tide returns to Hamlin Pool, the reactivation of the dormant mat is a physical process before it is a biological one. The incoming seawater contacts the hardened, desiccated mat surface. Water molecules, polar, hydrogen bond forming, with an extraordinarily high affinity for the charged functional groups of the EPS matrix, begin to diffuse inward through the crust. They insert themselves between the cross-linked polymer chains of the EPs, competing with and displacing the ion bridges that had locked the matrix into its rigid, dehydrated state. The crust doesn't fracture or crumble. It softens from the outside inward as water rehydrates the polymer network layer by layer. This process requires no metabolic energy from the dormant cells. It is driven entirely by the thermodynamic affinity between water and the hydrophilic regions of the biological matrix, a spontaneous decrease in free energy as water molecules distribute themselves into the system according to their chemical potential gradient. As the EPS matrix rehydrates, the osmotic pressure around the cells normalizes. Ionic concentrations drop back into the physiological range. The chaperone proteins holding the cellular enzymes in their dehydrated conformations release their grip as hydration is restored. The cellular membranes, held in a rigid, compressed state during desiccation, regain their fluid, phospholipid bilayer character. Ion channels reopen. Electrochemical gradients reform across the membrane. The metabolic machinery begins again, not by being rebuilt, but by being released from suspension. The enzymes were never denatured. The DNA was never fragmented. The structural proteins were never displaced from their correct positions. Because the EPS matrix maintained the spatial organization of every cellular component throughout the desiccation event, the return of water restores function without requiring repair. The cyanobacterial filaments extend outward from their dormant configuration. The photosynthetic apparatus orients toward the light. Carbon fixation continues. EPS secretion carries on. The daily vertical migration proceeds. The mat returns to its active state, ready to build the next lamination. Add the next daily layer to the archive. Trap the next grain of sediment, exactly as it has done every day for 3.5 billion years. The stromatolite's survival across geological time is not a consequence of competitive dominance, rapid adaptation, or metabolic versatility. It has survived by being, in a thermodynamic sense, boring. Its metabolic rate is low. Its growth rate is low. Its structural complexity is minimal. Its chemistry is ancient and conserved. It occupies environments, hypersaline, UV-exposed, periodically desiccated, that most organisms cannot tolerate. Not because it has sophisticated mechanisms for tolerating them, but because it has refined its chemistry to the point where these conditions are simply part of the cycle. Desiccation is not a crisis. It is a phase. The mineralized architecture of the stromatolite means that physical stress, pressure, temperature, UV exposure, chemical fluctuation is absorbed by the stone rather than transmitted to the biology. The organisms live within a geological structure of their own making. The outside world encounters the stone. The inside world encounters stability. This is the principle that runs through the entire fossil record of the stromatolite. Permanence in biology is not achieved by resisting the environment. It is achieved by aligning with the physical constants of the environment so precisely that the environment has nothing to act against. The stromatolite is not fighting the hypersaline water, the tidal exposure, the UV radiation, or the passage of time. It has made itself indifferent to all of them. The oldest multicellular animals are approximately 600 million years old. The oldest vascular plants, around 430 million years. The oldest birds, roughly 150 million years. The cyanobacterial mats of Hamlin Pool precede all of them by a factor that makes their histories feel like recent news. They were here before the first fish. They were here before the first cell with a nucleus. They were here before the oxygen in the atmosphere. They are still here. The tide has come in over Hamlin Pool. The mat surface, desiccated through the afternoon, is rehydrating. The cyanobacterial filaments are extending. The thylakoid membranes are orienting toward the last of the evening light. The EPS matrix is softening. The daily lamination continues. The stone beneath is 3.5 billion years of the same process, layer by layer, archived in aragonite and calcite, indifferent to everything that has passed above it. Out beyond the foray sill, the Indian Ocean is doing what it has always done. The desert behind the shore is doing what it has always done. The mat is doing what it has always done. Your body knows how to do what it has always done. The mat is building. The tide is steady. The archive is undisturbed and will continue to be, hardening quietly, layer upon layer, doing nothing interesting at all, which is precisely the point. The stromatolite has been boring for 3.5 billion years, and yet it is still here. Good night.